Hello and a very warm welcome to everyone joining us for today's Select Science webinar entitled Label-Free Techniques for Drug Discovery, Surface Plasmon Resonance and MALDI Mass Spectrometry for Primary High-Throughput Screening and Secondary Screening. My name is Dr. Lois Manton-O'Byrne and I will be moderating today's presentation. I am delighted to introduce today's speakers, Micah Hamister, Director of Small Molecule Pharma Business at Bruker Daltonic, and Sven Malik, Senior Application Scientist at Bruker. Just before I hand over to them, I would like to mention a couple of things. Please feel free to ask any questions for the Q&A session at any time during the webinar by clicking on the Ask button at the top right of the auditorium. You can view the presentation in full screen by clicking on the tab at the bottom right of the window player, and the on-demand link will be provided after the webinar. Without further delay, I am delighted to hand over to Micah and Sven, and I'd like to thank them for presenting to us today. Please go ahead, Micah.
Thanks a lot, Louis, and welcome everybody to our webinar about high-throughput screening, or even ultra-high-throughput screening with mass spectrometry, and to introduce our new surface plasmon resonance instrument, the SIERRA SPR-32. SIERRA Sensors, based in Hamburg, Germany, was acquired by Bruker just on June 4th this year to complement Bruker's label-free ultra-high-throughput screening mass spectrometry solution, the rapifleX MALDI PharmaPulse, which is subject in the first part of the webinar. So, I'm advancing the slide now. And here we are. Bruker Daltonics offers a leading portfolio for drug development. The latest is the SkyMax MRMS instrument with more than 20 million resolving power, the timsTOF Pro Ion Mobility Spectrometer, and of course the rapifleX MALDI TOF or TOF, which is the heart of our rapifleX MALDI PharmaPulse for high-throughput screening. The very latest addition is the before-mentioned surface plasmon resonance instrument, which came with SIERRA Sensors to us, and which is the focus of the second part of our today's webinar. Here we are. Definitely, one of our core technologies is MALDI Mass Spectrometry. Last year, we celebrated 25 years of MALDI Mass Spectrometry, and I'm not exaggerating when stating that there is a passion and dedication in the company to develop this technique ever further. Bruker has a fleet of MALDI instruments for different applications, from small benchtop machines, the workhorse AutoFlex, the high-end UltraFlex machine, and we have finally set a new standard with the introduction of the rapifleX MALDI TOF instrument, which is also the base of our rapifleX MALDI PharmaPulse for high-throughput screening. The rapifleX is the only available MALDI instrument using a 10 kHz laser system. And we are very proud now that we could add surface plasmon resonance to our label-free high-throughput screening portfolio, and that we are the only company that owns mass spectrometry, NMR, and surface plasmon resonance, and that SPR is now part of one of our key initiatives to develop label-free technologies. With the rapifleX MALDI TOF, we have developed a completely new MALDI mass spectrometer, which is able to screen more than 1 million samples per week at lowest cost. This is basically the story of our new rapifleX MALDI PharmaPulse we like to introduce you today. Before I start, I very quickly, for those of you maybe never heard of MALDI-Time-of-Flight mass spectrometry, in time-of-flight mass spectrometry, or TOF as we say, ions mass to charge ratios are determined by the difference in time they need from the source to the detector. Thinking of high-throughput screening, the advantage of time-of-flight mass spectrometry is definitely fast acquisition. As a non-scanning instrument, it acquires full mass range spectra without sacrificing speed. MALDI stands for Matrix-Assisted Laser Desorption Ionization and is used as the ion source for our time-of-flight mass spectrometer. The sample is embedded in a light-absorbing matrix, for example 4-hydroxy-alpha-cyanocyanamic acid, followed by excitation of the matrix molecules by laser light. And finally, ionization of analyte ions. Actually, this principle was awarded with a Nobel Prize in 2002. You can imagine that the laser, which is used for ionization, plays a major role, but more about that shortly. But let's talk about typical applications of MALDI-Time-of-Flight mass spectrometry. Typical MALDI applications are top-down sequencing, protein characterization, molecular imaging, glycoanalysis, and so on. Relatively new on this pie chart is actually high-throughput screening. So why was MALDI not applied earlier for this application? Very simple explanation. It was just too slow. The design goal of the rapifleX MALDI TOF was much higher speed, significant improved robustness, and overall just better performance than ever seen before. R&D scientists love this kind of specs. In result, the rapifleX was developed from scratch and sets a new standard in MALDI time-of-flight mass spectrometry. However, this was mainly to accelerate imaging applications. Now I'd like to show you two examples which makes this very clear. On this slide you can see a sample measurement with the rapifleX TOF on a rat lung tissue. So an imaging experiment. 400 laser shots on each spot. It took 41 minutes. With previous instruments using our 2 kHz laser, it would have taken about 13 hours. The vision, why not use it for HTS, was mainly inspired by our MALDI customers from the pharmaceutical industry. Of course, even it is the same machine, to develop the rapifleX from imaging to high-throughput screening, we had to solve a couple of, so to say, challenges, which are listed on this slide. As you saw on the imaging example, it does take not more than 10 hours anymore to do an imaging experiment, but still about half an hour. In consequence, sample plate exchange is manually. A 1536 sample plate takes between 8 and 10 minutes, so about 1,500 plates for a 2 million sample screen. Manual exchange is no option anymore. Therefore, we have developed a new sample feed mechanism which allows fully automatic sample exchange by robot systems. Other issues were sample plate preparation, assay development, unattended operation for millions of samples, and of course, cost per sample not to forget. Finally, we had to develop a new software suite for high-throughput screening. As said, main application we had in mind when developing the rapifleX was imaging at that time. Throughput for imaging increased by a factor of about 20. The rapifleX, with its new laser and robust ion source, allowed constant operation. By the way, this is a molecular image of a rat brain. Now it was an obvious move to go from imaging to high-throughput screening. For the mass spec, it doesn't make a difference if analyzing a tissue sample or a high-density spotted sample plate. Actually, a 1536 sample plate is much less demanding. Almost no material enters the instrument. The sample volumes used for spotting are in the nanoliter range. To conclude, it was developed for imaging but meets all requirements for ultra-high throughput screening. Here you see a MALDI sample plate with 1536 samples. Each sample spotted with 100 nanoliter. With this slide, I like to visualize the difference between a 2 kHz laser system and the 10 kHz laser system as we have it in our rapifleX. As I pointed out earlier, the laser, which is used for ionization of the samples, plays an important role. Our other MALDI models use 2 kHz laser, which is already very fast. You can see that on the left side here in the little video. However, our new rapifleX uses 10 kHz neodymium YAG laser. Those lasers are especially developed by Bruker for most efficient use in MALDI sources. The so-called smart beam profile is designed for optimal scanning of MALDI samples. Maybe it is difficult to imagine what the difference between 2 and 10 kHz laser means for scanning of samples. Therefore, I'd like to show you the little video now for the 10 kHz, which you can see on the right side now. Probably you will immediately think, but this is not just 5 times faster. And yes, that's true. The rapifleX has not just a faster laser, the whole process of sample scanning is optimized. But one of the key features is the scanning laser. Compared to our other MALDI instruments, the rapifleX moves not just the stage underneath the sample, but scans the laser by a system of mirrors. This is what we call the scanning laser. But enough of technical details. Let me explain why high-throughput screening of biochemical assays in drug discovery can benefit from mass spectrometry. In general, a mass spectrometer offers the advantage of label-free screening of substrates and products in enzymatic assays. Labels are not required because the mass spectrometer detects a difference in mass which occurs when an enzyme is inhibited or not by a molecule, by a compound. It simply measures the disappearance of a substrate and the appearance of products. Other advantages are: it is a highly sensitive method, makes the development of fluorescence labels or antibodies redundant, and consequently accelerates assay development, minimizes the risk of interferences, and results in low cost per sample. Typically, mass spectrometry was not used for very large or primary screens of millions of compounds. Just when the number of compounds becomes smaller, especially solid phase extraction mass spectrometry, SPE, and also MALDI mass spectrometry came into play. Why that? The answer is again easy. So far, mass spectrometry was too costly per sample and much too slow to screen large libraries. So typically, fluorescence-based techniques are used for large HTS screens when hundreds, thousands, or millions of samples need to be screened. As mentioned before, mass spectrometry is typically used at later stage for confirmation screens of hit. So if, for example, a campaign with 2 million samples results in 40,000 hits, many might be false positives and need hit qualification by mass spectrometry. Mass spectrometry offers the benefit to measure the substrate product ratios label-free and avoids artifacts caused by the labeling of the compounds. So the obvious question arises, why not using mass spectrometry at the beginning to do it right the first time and to make lengthy confirmation screens redundant? Should advance in a second. Here it is. But coming back to the question of automation, the rapifleX MPP measures a 1536 sample plate in about 10 minutes. The new rapifleX MALDI PharmaPulse is equipped with a high-speed automatic sample loader which can be fed by robot systems. This slide shows a layout from SciBio Analytic Jena with robot, target hotel, and spotting device. However, we work with other automation partners like Hi-Res Biosystems, Beckman, Thermo Automation, to mention some. You should see the next in a second. We are looking for slide 16. No, I push it again. Here we are. Sorry for the delay. In this context, I'd like to draw your attention to a brand new paper from this month, actually. This paper from the HTS group of Boehringer Ingelheim describes their automation concept for their rapifleX MPP, including reformatting from 384 to 1536 format, and demonstrates sample measurement times of 0.4 seconds per sample for MALDI analysis and 0.6 seconds per sample for preparation of the 1536 sample plates. Impressive Z values between 0.08 0.87 and 0.91 could be obtained. When it comes to sample plate preparation, things to consider are speed, of course, so that the spotting procedure is aligned with the speed of the rapifleX and meets the throughput demand of our customers. The spotting device should allow nanoliter volume handling. We typically use 100 nanoliter and, of course, high precision and accuracy, no cross-contamination, and ideally a washing step to eliminate, for example, high salt concentrations, which are not in favor with MALDI ionization. Most custom automation solutions of our rapifleX MALDI PharmaPulse use today the SciBee well from SciBioAnalytic Jena, which offers beside a 384 head, and I show a close-up now, a 1536 head with really 1536 tips, as you can see on this slide now. Advantage is definitely the speed and the possibility to desalt the sample. Obviously, fluorescence assays needs to be changed to label-free assays, and things like buffer and salt concentrations need to be adapted to MALDI Mass Spectrometry. Meanwhile, several papers are published about the adaptation of assays for MALDI. A couple of examples are shown here. The perfect assistance for assay development is, of course, the perfect assistant for assay development is, of course, the rapifleX instrument itself, and therefore we have developed a module within our HTS software which automatically finds the best conditions. Typically in 384 format, different assay compositions, dilutions, and instrumental conditions will be combined, and within minutes the rapifleX identifies and prioritizes the best assay. With MALDI HTS, not just the product masses of an enzymatic reaction are being measured, but also the substrates. Current setting is up to 30 masses. I say currently 30 masses because since the MALDI was never as fast, new applications arise all the time. And I'd like to draw your attention to a brand new paper from Merck Kenilworth in the USA, which used their rapifleX MPP for even larger synthesis screening and measure hundreds of masses per plate. So let's talk about money now. From the beginning of this journey, cost per sample was one of the most important parameters. MALDI in general offers low cost per sample because it uses very low volume, nanoliter, no labels, no solvent, no tubes, columns, and so on. The largest contributor were the MALDI sample plates, which are suitable for many MALDI applications, but they were too heavy to be handled by robots and far too expensive. Meanwhile, we have a fit-for-purpose disposable HTS MALDI plates and could reduce the cost per sample, for example, for a 1536 format to about 3 cents per sample. Of course, the more you spot, the lower the cost, because the plate is the main contributor. Hope it works, yeah. To quickly summarize, the possibility of very fast label-free analysis by mass spectrometry is definitely key for the success of the MALDI PharmaPulse, since it avoids artifacts from the labels. It is very sensitive, uses nanoliter sample volume, and doesn't need solvents. Consequently, cost per sample is very low. For the first time, mass spectrometry can be used for large biochemical screens. Champion data are less than 8 minutes for a 1536 plate. If you're interested to read more about it, I like to recommend the SLAS Discovery issue from December 2017, which was dedicated to MALDI Mass Spectrometry and contains, among other MALDI papers, articles by several rapifleX MPP customers like Merck, Genentech, GSK, and University of Dundee and Newcastle. Here's, for example, the paper from Merck Darmstadt about kinase screening. From Genentech in San Francisco doing non-covalent binder screening. And another kinase paper from Rachel Heap of Matthias Stroh's group at the University of Newcastle. But now, next slide first. But now enough of MALDI PharmaPulse, and I'd like to hand over to my new colleague Sven Malik to explain our new SIERRA SPR-32 to you. Sven is sitting beside me, and I will just hand over the micro to him now.
Hello everybody, and thanks for your attention. Today I want to give an introduction of the SIERRA SPR-32 and show you that controls define throughput. The SIERRA SPR-32 is the latest SPR version in the Bruker system portfolio. The system has an increased throughput due to its 32 sensor spots, and with robotic integration you can even increase that throughput. So you will be able to screen up to 14 microtiter plates unintended. Compared to competitor systems, you will have higher performance, greater flexibility, and a more robust system. So let's go into detail. In terms of throughput, the system itself has a 8-needle/channel sample processing unit that you can see in one of the images there with a needle head. The system itself has a dual plate deck, and per channel you will have 4 sensor spots, so in total you have 32 sensor spots that can individually addressed. In terms of flexibility, you will have the possibility to individually control a single channel, so you can have or use the system for assay development just on a single channel, and once you have the optimum parameters, you just use these parameters for all of your 8 channels. Additionally, you will have the possibility to do a 4-buffer analysis, but more details on that later. In terms of performance, you will have a sensitive detector that is able to detect low molecular weight compounds down to 100 Dalton. And additionally to that, you have a robust microfluidics that can handle 70% supernatant, for example, without clogging. And we even have customers that inject show cells through the microfluidics without clogging the microfluidic membranes. In terms of the key points that I'd want to focus on within my presentation, you see them listed here. So it's the 4 sensor spots per channel, it's the 4-buffer analysis, it's the flexibility and performance of the system, and additionally the sensitivity. So let's focus now on the 4 sensors per channel and how we address our sample to the microfluidics. We use a patent which is called Hydrodynamic Isolation, and as Mike said, this patent comes from SIERRA Sensors that allows to introduce and inject any sample, any sensor at any time. And in order to explain our technology, please focus now on the cartoon. Within the microfluidics, we have no membranes or microvalves as competitors. We just have fluidic streams that we introduce into each other, and how we do that you will see in a minute. Per channel you will have a buffer inlet port and a waste. Per channel you will have so-called sample inlet and sample outlet ports, and when you dock a sensor you have the designated sensor spot locations as you can see here in the cartoon. and how we address our sample. If you focus now on the image detail, per channel we have a continuous flow buffer pump providing the buffer into that channel, and then we use a sample pump injecting into the buffer stream. And depending on the addressing that the user is choosing, we can individually address just a single spot, as you see here in the example, or you can address multiple spots at the same time. This flexibility allows the customer to use the individually addressing to create the sensor surface, whereas the multi-spot addressing can be used to within the experiment itself. What you see here on the right-hand side of the slide is a live camera image that you will also see within the control software. So we have an SPR imaging detector combined with a high sensitive, so it's a high sensitive SPR imaging detector, and that live camera image will be provided additionally in the control software. And you count from left to right the channels 1 to 8, and from top to bottom the spot locations. So you have A, B, C, D. These are the spot locations within the channels 1 to 8. And as I said, you can individually address the spots A, B, C, or D. You can address them together, A, B, C, D together, or just a triple pair, A, B, C, B, C, D, and just a dual pair of A, B, B, C, and C, D. That means you're fully flexible on the sensor design, and you're fully flexible in terms of usage of your consumables. So how could that look in an experiment? So for example, if you use small molecules, or if your application focus is small molecules, what you could use the system for is that your target is immobilized on all D spots of the system. Then you have a mutant immobilized on the C spot, and maybe a reference protein on the B spot, and just the active blocked surface on the A spot in all of the 8 channels, and you work in parallel. For the application of antibodies and proteins, you could test in parallel for cross-reactivity. You could test multi-target, as you could load up to 31 ligands to your sensor surface and keep just a single sensor spot as a reference. One of the applications that is used in the industry is already the epitope mapping, for example. So you could inject, due to the 8-channel and 8-needle sample processing unit, you could inject 8 samples versus 4 targets on the sensor surface per channel. Within the system, and due to the microfluidic pattern that we use, you have a excellent sensor reproducibility. As an example, I have here a typical immobilization sensorgram where you can see the immobilized protein and the RU signals that we got, plus the standard deviation over all of these channels. Plus, due to the fact that we have a fast transition between the events of running buffer, sample, running buffer, you can detect fast on and off rates if your molecules bind that fast or have a fast off rate. And the third fact is that there's no carryover between the individually addressed sensors. Means if you address just the B spot, as you see it here in the live camera image example, there's no leakage to the spot C and D in the same channel. And what does that mean for the throughput? What you see here is an example where we compared a single needle system with a 6-needle system with an 8-needle system. And this is in a screening comparison of 2 96 well plates where we had an on rate of 2 minutes, an off rate of 5 minutes, and a 2-minute regeneration. And this just shows the throughput of these 296 well plates. Per day, you can, and this is just an example, of course, that depends on your assay parameters, but per day you can screen 3,000 more samples and get 10,000 and more control-subtracted interactions. A typical example, and if we talk about screening, this could be a typical example, or is a typical example that is used in the industry already. So, as I said, the target surface is in all D spots in all channels. The reference surface with a reference protein is in all C surfaces, C spot surfaces in all channels, and just the activated blocked surface will be in the B spot. And then what the customer in this case has done is they were screening 384 samples, and they had each of these small molecules at a concentration of 100 micromolar and ppb in a buffer that contains 3% DMSO. So we were running 3 times the DMSO calibration curve at the beginning, in the middle, and at the end of the whole plate. And in totally, it takes 3 hours, close to 3 hours, to measure 384 samples. And if you want to increase that throughput, you can just hook up a robot to this system. And this feature you see here, so this is a photo that I included from our demo lab in Hamburg, where a robot is connected to the system directly and feeds the system while it is to increase the throughput. And so the system itself has a dual plate deck, plus the robot you will have 12 plates more. That means in total you can screen 14 plates unattended. And in this example we used an Orbiter RS from Thermo, but any robot can be used. And the feeding of this system with the plates is of course fully software controlled. And for the potential customers that use and screen protein-protein interactions or work with protein-protein interactions, of course, each of these plate locations can be temperature controlled using a chiller. One of the features that comes with the system is the 4-buffer analysis. And this is the so-called buffer box. It contains a degassing unit and it contains a set of 4 buffer syringes, whereas a single syringe supplies 2 channels. That means the user can use up to 4 different buffers in parallel and check for the impact of cofactors. So just using a click in the software, you can either use the same buffer in all channels, so if you have the optimum parameters within your experiment, or you can just use it with 4 different buffers in parallel to check for DMSO concentrations in the buffer, to check for different salt contents, check for target stability, and as one of the applications you can even use it to do some kind of cofactor analysis or cofactor side effect assays, where you see how the off rate and on rate is changing depending which cofactor is used. In terms of the flexibility and performance, one of the key features that we provide with the system is the individual needle control. This feature allows the user to use the system as a development tool to check for parameters, and once the optimum is found, you can use it as an 8-needle high throughput system. And what the benefit of these features is that you could reduce your sample consumption and optimize the sensor life and the usefulness of the consumables. So you save on time and you save on consumables, as you can of course reuse the sensor surface and you just increase the flexibility using that tool. And what we just have, independent whether you decide to use a needle or not, you can have solution in the vials or not. It will just pick up from the needles that you told the software, that you told within the software. And this just increases the flexibility and again the usefulness of your consumables. And this is an example where a customer used that. So here in that example, if you focus on the live camera image, the first 4 channels were already used with protein and the immobilization protocol. So the first 4 sensors have already protein immobilized, and then the customer decided just to use channel 5 and 8 in parallel. So you see that the live camera image that you see at the bottom, this is a typical addressing or a typical image that you would get for the activation of the surface and the deactivation. In this case, we just addressed the sensor spots A and B in channel 5 and 8. And for the protein injection itself, we just injected on sensor spot B in channel 5 and 8. And if you see the high refractive or the signals itself, if there would be any leakage to the sensors below, you would see that there's a signal coming up from the baseline as the signal of all 32 sensor spot is collected at all time. In terms of performance, we recently launched a new feature in our new control software, which is called Frame Inject. This feature can be used to do mechanic studies of your interaction and also use it for cofactor analysis. And how it works is that you use a so-called frame solution. It's a frame surrounding the core solution or core inject, and you can use, and that frame solution or frame buffer can be completely different from your system buffer. So this is a nice feature we had recently or is recently used. So let's talk about the detection sensitivity of the SIERRA SPR-32. We use the SPR Plus detection, which is a patent from Agilent, and when I'm right, the publication was from 2006. And what they do, or what we do, as we have the same detector, we use a bright light source and a high-speed camera, and within that arrangement we have an acousto-optical deflector that deflects the light to the sensor surface. And the nice thing is that the acousto-optical deflector is synchronized with the camera, so each time the light is deflected to the sensor surface, the camera takes a picture. And this allows us to have a really, really, really low baseline noise compared to other imaging systems, and I will show you data in a bit. So the baseline noise is lower than 0.02 RU. And what you see here now is an example for that sensitivity. So this is 2 RU data of small molecules that range around 200 Dalton and 150 Dalton. So you see that you can distinguish easily between the off rates of these 4 compounds that are injected. Another example is shown here where we just did a thermodynamic study. So we injected the same concentration of analyte at 13 different temperatures and just observed the interaction, and you see that there's a change in the off rate due to the temperature. So when you increase the temperature, the interaction gets faster, or the faster is the off rate. But also for the protein-protein interaction, you will be able to detect low RU values, as you can see here within that example. Let's talk about the consumables. As I said, the system itself without robot has a dual plate deck, so you can use standard microtiter plates, 96 deep well, and of course also 384 well plates. Additionally, what you have is a reagent trough that can either have 2 times 19 or 1 times 40 milliliter of solution, so you could use it for a regeneration, for example. But also you can have a 3 times 8 vial holder. On the sensor itself, we have standard sensors like the amine flat or the HCA sensor, but also his-tag capture sensors or biotin tag capture sensors. Any sensor that you are using with your current SPR setup and we do not provide, you can get from Xantec. If you just take a second and you look into your lab and you compare the current systems that you have in your lab, then I might help you to find yourself here within the SIERRA SPR-32. A system that has 4 flow cells you see here. So with the SIERRA SPR-32 you have that 8 times on a single sensor, and a system that has 16 sensor spots that you have 2 times on a single sensor with the SIERRA SPR-32. So the system itself is economical as possible. A definitely important point is of course also the software, and we are launching on the way to launch that software to all of our current customers. It's the new R3 software, and I just show you some examples. So we focused on intuitive design. So we have a status bar at the bottom, and the design of the software itself should be or is similar to an application on your mobile phone. And the software can be used with a touch screen, so it's intuitive and it's similar to your mobile again. So you just click and play. You will find pre-written methods throughout the whole experiment. So the preconditioned immobilization and the experiment itself, we provide as the application team, we provide pre-written methods, but of course you can also write up your own methods within the assay development tail. We provide the possibility of user level hierarchy. That means group leaders provide and write up methods, and the user itself can just exchange parameters or change parameters like the flow rate or association time, and set up the data storage path, for example, but cannot change the method itself. And as an example of the software and the flexibility itself, so this is just an example. So we implemented a pre-analysis already in the control software, and as you know, not everything goes well. And for example, if your designated reference spot is sensor spot 3A, and due to an error you cannot use sensor spot 3A because you did a mistake with the pipetting or whatever, and what you can do just on the fly is changing that setting, and you can just use sensor spot 2A as your reference spot. And here what you see, you can on the fly just show and check what the signal looks like if you use that reference spot within the control software. And also, if you use the feature of the individual needle control, you can on the fly check and uncheck sensor spots and channels that you want to see or do not want to see. So let's come to the summary now. The SIERRA SPR-32 is faster as competitor systems, as the control spots are already measured. So there's no rerun of experiment necessary because you have all information in one experiment. You have 32 sensor spots in a 8x4 array, and of course, depending on your experiments, you will be able to detect 10,000 interactions a day. You have an improved flexibility due to the fact that you can individually control the needles, and you can also use 4 buffers in parallel if you want to. You have the fragment sensitivity, as you have a ultra high sensitive detector within that system, so you can detect small molecules as you have seen in the examples, and you have a robust microfluidics. So again, people and customers using that, and they screen in 70% supernatant without clogging the microfluidics, as we have seen in our or as we have heard about competitor system, and even show cells were injected through the microfluidics without clogging the microfluidics. And you have an intuitive software that combines and combines that nice package of a flexible instrument and a flexible software. So I hope I was able to show to you what the SIERRA SPR-32 and where that system can help you in your experiments. And yeah, we are happy to give you a more detailed webinar on the software if you want, also with the analysis software.
Thank you. That's perfect. Thank you both for such an interesting presentation. Now let's move on to the last part of today's webinar, the question and answer session. There's lots of fantastic questions coming through, so thank you so much for that. I'll just start off with the first one. What is included in your cost per sample calculation for the MALDI HTS?
Thank you, Louis. I think that's a question for me. Here's Maike. That's a very good question, and I was maybe not very clear on that. I mentioned in the first 1536 format, it's about 3 cents. What's included in that is actually all which is important for the rapifleX itself. For example, I mentioned that I think the main contributor is really the sample plate. But also the MALDI matrix, what you need. But also in addition, all the bits and pieces you need, so the consumables and the, but also spare parts. You know, we have included that all already into the 3 cents per sample.
Fantastic, thank you, Maike. Next question. Is it just possible with your SPR to address all spots per row at the same time?
So per injection you can address either all spot at the same time, or you can use the individual needle control to just send across the sample to a designated spot location in designated channels, and this is easily done within the control software. So you just check the channels you want to use and you check the addressing that you want to do.
Great, thanks, Sven. Is Bruker working with a specific automation vendor to automate the rapifleX?
Thanks. That's definitely for me again. Yeah, and the answer is clearly not. I mean, we have actually started and thought about to do that, but it came out that most customers of ours, they had their preferred vendor for automation. So we decided finally to go with an open interface. We described that, we support the automation vendors, but yes, it's an open interface, and you can use. I mean, so far we have not met an automation vendor which we cannot accommodate for the rapifleX. So yeah, you can use your preference for that. Mm-hmm.
Great, thank you. Does the SPR system have a degasser?
Yes, within that buffer pump there's a degasser built in, and you got the option either to use, as I have shown in the presentation, either use 4 buffers in parallel or using the same buffer in all 8 channels.
Great, thank you, Sven. What is the maximum capacity of ligands per sensor with the SPR?
So the question gets then also to me, I guess, yes, because it's SPR. So in maximum, you could have 31 ligands on the sensor surface and have a single sensor spot as reference, and then send across the same antigen overall in all 8 channels. So that means 31 active sensor spots and 1 control. So this is what people do. For example, if they want to do an off-rate screening, they use this approach. But if it goes to kinetics, so what we suggest is have then per channel an inline reference spot. So as I have shown in an example, so we would have the reference on A or B, and then furthermore controls. Yeah. So this is what the customers are using it for.
Brilliant, thank you. You mentioned imaging for the MALDI rapifleX. Is that possible with the same instrument?
So that's for me again. Yes, indeed. The imaging and the HTS and all the other applications you saw is possible with the same rapifleX instrument. So you have that in your lab. I mean, many customers use it dedicated for HTS or dedicated for imaging, but you can use the same machine. These are basically different software modules. So it's a very flexible machine. Thank you.
Fantastic, thank you. We've had lots of fantastic questions coming through, so thank you so much to everyone. We'll just try and get through a couple more, but I know there's lots more in the system, so we'll work our way through those as well offline. What is the required sample volume per spot with MALDI?
Okay, that's for me again. Yeah, I mentioned normally we use 100 nanoliter sample volume per spot. It can be also more. Of course, they shouldn't, in 1536, they shouldn't touch. But we can even go lower. We have some customers experiencing for even higher throughput with sample geometries of 6,144, and then we actually reduce the volume to 20 nanoliter. Of course, everything is done with a spotting robot. So let's say between 20 and 100 nanoliter is quite normal.
Great, thank you, Maike. Do you support features like ABA or single cycle kinetic?
Yes, so it's the same feature, but we have a different name, of course. So the ABA is, if you remember, that is so we call it frame inject. It's exactly the same feature, and we have it introduced recently, so definitely that works. And for the single cycle kinetics, definitely that is also possible, and our customers are using it that way. But an alternative approach is to have the concentration series over all 8 channels, as then each channel gets a certain concentration, and you get the on and off rate for all of the concentration in your whole concentration set. So that would be an alternative approach. We call that single injection cycle kinetics. Yeah, but yes, we have. So it's definitely yes, we have both features.
Great, thank you, Sven. We've just got time for one more question. What is the difference between the small molecule binding MALDI approach published by Genentech and what you can do with the SPR?
I can answer definitely for the Genentech paper, and this is really a very interesting paper, because normally you would think non-covalent binders, so you can't screen with MALDI. But Genentech uses modified plates from so-called SAMDI plates from SAMDI Technologies. So it's another vendor of MALDI plates, which functional surfaces, and they enable that you can screen non-covalent binders. So I really can recommend the paper. It's a long story to say it in a minute, but read the paper and you will see the power of combining this SEMD technology with MALDI for HTS. Thank you.
Fantastic. Thank you very much. Well, that's all we've got time for today. Thank you very much to Sven and Maike for today's informative discussion and presentation. Thank you to everyone joining us online and for your interesting questions. I hope that you have found this a worthwhile session. If we didn't manage to answer your questions, we will follow up with you personally. If you have any more questions, please feel free to email me at editor@selectscience.net. If you would like to listen again to today's webinar or invite a friend to listen, it will be available on our website shortly. Goodbye, and thank you once again for listening.